This video is continuing the cell chapter. Characteristics of cells. Cells vary in shape. You have cells that are oblong, cells that are more square-like, more that are circles. And they are specialized in that they perform a specific function, such as red blood cells carry oxygen, white blood cells protect against foreign invaders, bone cells deposit calcium and form the skeleton of your body. Nerve cells conduct electrical impulses, and skin cells protect against infection. So they all have a job to do. And there are two major regions of a eukaryotic cell. You have your nucleus and you have the cytoplasm. So here is the nucleus in this cell, and you have the nuclei, plural, in these cells here. So it's pretty distinct. It's usually the big circle that's inside the cells. And then you have the cytoplasm, and that is the space outside of the nucleus, but still inside the cell membrane. So once again, our nucleus, that's where we're going to go next. The plural, if you have more than one nucleus, it'd be nuclei. And we learned it back a long time ago as the control center of the cell. It's located most often at or near the center of the cell, and it's surrounded by a nuclear envelope. So the nucleus has its own membrane, and it has a bilayer or double membrane of phospholipids as well. There's also perinuclear space between those two membranes, which you guys saw on your diagrams. The nucleus contains the nucleoplasm, which is the colloid, very similar to the cytoplasm, but it's inside the nucleus instead of outside. Inside the nucleus, we have two types of organelles. You have your nucleolus, which is that purple thing we're seeing over here. It's another dense circle within your nucleus. And it is not surrounded by a membrane, so it is not membrane bound. Sometimes there's between 1 to 4 per nucleus, so you could have more than one nucleolus. Inside of the nucleolus, you have RNA and protein, and its job, the nucleolus's job, is to make ribosomes. So we're going to learn a little bit about ribosomes in a little bit. The second thing that's inside the nucleus is genetic material, which is DNA, and that will exist in two forms. You've got chromatin, which is loosely coiled threads of DNA, and they're wrapped around proteins called histones, which you see here. The DNA would be the orange-yellow string, and you can see that the purple, these are the histones. You can see they're wrapped around these histones like a spool of thread, if you will. Then you have chromosomes, which are tightly coiled rods of DNA wrapped around proteins called histones. So they're very dense and compacted. Most of the time when you see them, they look like X's. So the difference between these two things, they're the same thing essentially. They're just organized differently. So chromosomes are very tightly packed, and chromatin is relaxed and loose. Examples to try to compare it, chromosomes are like your pack of ramen when you get it out of the package. It's all hard and condensed. But your chromatin is once you've cooked the ramen, the noodles spread out, they relax, they're not as organized. And DNA functions in heredity and protein synthesis. So we will have a whole chapter coming up on DNA, RNA, and proteins in a little bit, a couple chapters. The nucleus has nuclear pores, which you can see here on the diagram. They're little holes basically in the nuclear membrane, and that allows materials to pass in and out of the nucleus. Information typically to go from the nucleoplasm to the cytoplasm. All right, the next part is the cytoplasm itself, and this is the colloid inside the cell. It's made up of protoplasm, and that's the most important colloid in nature that we talked about last chapter, I believe, or the chapter before. And it can undergo phase reversal, and another name for phase reversal is cytoplasmic streaming. Another name for cytoplasmic streaming is cyclosis, and that's just, once again, phase reversal, changing from a sol to a gel or a gel into a sol. You will have a question on your test about phase reversal and another other name for it. So the answers would either be cytoplasmic streaming or cyclosis, so make sure you take note of that. We know that the Two causes for cyclosis could be the change in temperature or pH. The cytoplasm is surrounded by the cell membrane or plasma membrane, which is thin and flexible. So oftentimes we think of the cytoplasm as this empty space within the cell, but it's not. It is a colloid, and everything, all the other organelles that we're going to talk about, are floating within that cytoplasm. So talking about the cell membrane here, we're going to talk more about the cell membrane in our next chapter. And we talked a little bit about it last chapter. We know the cell membrane is made up of phospholipids, but as you saw in your diagrams, it could also have infoldings or outfoldings. The infoldings are called involutions, and the outfoldings are called microvilli. Cell membrane is said to be semi-permeable, which means it is selective. It allows some things through, but not everything. It depends on what could fit through this layer and what needs to go through these protein channels, which once again we will talk about next chapter. Once again, we have two phospholipids. The hydrophilic heads are oriented towards the watery cell and the outside watery environment. And the hydrophobic tails face the interior of the cell membrane where there is no water. The cell membrane does have proteins embedded in it, which we'll talk about more next chapter. They aid in getting materials across the cell membrane. Because the membrane has these proteins in addition to lipids, the cell membrane is often referred to as a lipoprotein membrane. So lipo for our phospholipids and protein for our proteins that are embedded here within the cell. Okay, our next organelle we're going to talk about are vacuoles. And vacuoles are membrane bound, so they are in eukaryotes and not in prokaryotes. They are found within the cell's cytoplasm. And what their job is they hold a variety of substances. They hold food, waste, oxygen, and also carbon dioxide. So they are the storage center of the cell. So I like to think of a vacuum. When you use a vacuum, all the dirt goes into a canister or to a bag, and it will hold that material until it's full and ready to, throw out. So that's how I try to think of vacuoles and vacuums. They are often formed by involutions of the cell membrane. So what happens is we have the cell membrane, and say this is a material that wants to get into the cell. Okay, it pushes in on this cell membrane. The cell membrane is thin and flexible, so it makes this little pocket called an involution. And the material will travel into that pocket, and then as it pushes towards the inside of the cell membrane here grows closer and closer together until it is able to fuse, and this little vacuole or pocket will pinch off of that cell membrane, and that is now a vacuole. Our next ones are lysosomes, and these guys are also sac-like structures surrounded by a membrane. They are formed by the Golgi apparatus. The Golgi apparatus will make these lysosomes. They contain powerful digestive enzymes that carry out hydrolysis. So their job is hydrolysis. So you see lysis here in hydrolysis, and they're called lysosomes. So their job is to break things down. Okay, they digest large food particles and bacteria. They break down damaged or unproductive organelles. So if like a mitochondria stops working or a vacuole or something, these guys will go and disassemble those unworking, not working organelles. They could also digest the entire cell, like self-destruct mode. If the cell is not working very well anymore, these guys will burst open, releasing all their digestive enzymes and kill the cell itself. So these guys, I like to call them the cleanup crew of the cell. So I think lysosomes, what is something that cleans, that starts with lice? Hopefully you're thinking of Lysol. So Lysol, lysosomes, lice means to break. Lysosomes are the cleanup crew of the cell. So here's a video for us that explains how lysosomes are made and what they do.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles such as old mitochondria. This results in the destruction and recycling of these structures.
So here is another website too, cellsalive.com. If you go on here, and you could actually look at an animal cell. It gets it's a little loud. But you can go through a nucleus and read about it, and then you could pick out other things. Okay, or you can return to the cell diagram. This might help you a little bit with identification if you need it. You can go to a plant cell. You have all the things in here as well. So you can check those out if you'd like. Okay. And I don't know what this is. We're going to check it out. Not found. Okay, not worried about it. All right, the next one, peroxisomes. Peroxisomes are yet another cell organelle. They act very much like a lysosome, except they work with something very specific. They work with hydrogen peroxide. So peroxisome breaks down hydrogen peroxide. They are found in most cells, but in larger amounts in the cells of the liver and the kidney. And the reason for this is that your liver and kidney produces hydrogen peroxide. There's that joke that I tell sometimes, if you've heard it. It says, a guy walks into a bar and he says, hey, I'll have some H2O. And the guy after him walks in and says, hey, I'll have some H2O too. Like also, the second guy dies. Because H2O2 is hydrogen peroxide. And if you drink this, you're probably going to die. It's not good for us to have. So our body does actually make this, and we don't want it. So peroxisomes will take that hydrogen peroxide and using the enzyme catalase will break it down into water and oxygen in this chemical equation you see here. Ribosomes are the next one. And when I say ribosomes, you say protein. Ribosomes, protein, ribosomes, protein. So protein is what the ribosomes do. It's its job. So this is something in 30 years from now when you come back and I'm an old lady teaching, you definitely want to know ribosomes make protein. Just like you know already that the mitochondria is the powerhouse of the cell. Now you should know forever that ribosomes make protein. All right. So they're numerous small and granular organelles, and they lack a membrane. So you're going to find these in prokaryotes and eukaryotes. They're made by the nucleolus, which we talked about already. And once again, their function is to make protein. Okay. Protein is formed by amino acids using DNA's recipe on the surface of the ribosome. They are protein factories, and we will be looking more at ribosomes in our DNA, RNA, protein chapter. They're found in 3 areas of the cell. You're either going to find them floating in the cytoplasm freely, on the rough endoplasmic reticulum, or on the outside of the nuclear envelope. Okay. Ribosomes of the RER in the nuclear envelope make protein for other cells, while the ribosomes of the cytoplasm make protein for that same cell that it's already within. So since we kind of talked about the RER with ribosomes, let's look at the endoplasmic reticulum. This is a complex network of interconnected membranes. This is the highway of the cell. It connects the cell membrane to the nuclear envelope and other organelles all together. It acts as an anchor for other organelles, and there are 2 types, rough and smooth. Okay, 3 functions: it transports substances, usually proteins, modifies substances, usually proteins, and also could store substances, once again, usually proteins. Okay. In our next video, stay tuned. We're going to talk about the Golgi apparatus.